T cells are part of the immune system and can be broadly divided in two major groups, the effector T cells and regulatory T cells. Now, the effector T cells have the function to recognize foreign antigen and to mount an immune response against this foreign antigen. Now, they do so because they express a very specific receptor on the cell membrane, and through this receptor, they recognize the antigen that is expressed by a very specialized antigen presenting cells, that is, cells that present the antigen to the effector T cells. Now, in contrast, the regulatory T cells, they have an opposite effect. They inhibit the immune response and they inhibit the effector T cell. And in particular, they are there to defend us from autoimmune attack. Namely, they just inhibit the response against autoantigen and against the antigen that are expressed by our own cells or our own tissue in the body. This means they have a very important function to defend us against the autoimmune attack. Okay, tRegs have a different way to control the unwanted immune response. They do through a soluble mediator, and they create an environment that is anti-inflammatory, or they do by target their effector cells by killing the effector cells, or they act through the antigen presenting cells that has the function to present antigen to the effector cells. And they do this in a way that these antigen presenting cells become incapable of presenting the antigen to the effector T cells, and mostly to activate the immune response. There are three different pieces of evidence for the role of tRegs in the immune system. The first is derived from IPEX syndrome patient. This patient have a mutation in one of the important markers for regulatory T cells, that is the transcription factor FOXP3. This patient, having this mutation in the FOXP3, develop multiple autoimmune diseases that affect the skin, the gut, different parts of the body, and also the pancreas, and they develop type 1 diabetes. Linked to this is a second evidence, and this derives from patients that have a different type of autoimmune disease, type 1 diabetes, rheumatoid arthritis, and what has been defined that the regulatory T cells, either the number or the function, is altered in this patient. And finally, and very much linked to the goal of Quell, is derived from animal model of autoimmune disease and transplantation, where the transfer of regulatory T cells create again an equilibrium, and they can either prevent transplant rejection or cure autoimmune disease. It is now the time for multiple engineered tRegs, because we know a lot more about the biology of regulatory T cells. We also have the knowledge that we can expand numerically regulatory T cells. And finally, and I think very relevant for Quell, that the molecular biology techniques, they really progress, and they allowed us to create these multiple engineered tRegs. Multiple engineered tRegs can be applied in many different inflammatory situations. They can be applied to prevent graft rejection. They can be applied for cure or prevent the development of autoimmune disease, but I think they have a broader application for any situation where there is an inflammatory response. There are different ways that we can improve the effect of regulatory T cells. The first is by rendering the regulatory T cells specific for the target antigen in a way that they can be localized where the target antigen is. The second is by improving their stability, and this, what I mean for this is that in a way that they will not switch from a regulatory function to an effector function, and lastly, to improve their persistence in a way that they can be around long enough to exert their effect. FOXP3 is a master transcription factor that is expressed by regulatory T cells, and like other transcription factor has the function to switch on and off other genes, and in particular in regulatory T cells render the regulatory T cells stable in a way that they will not switch to an effector mechanism, and also improve their function. By increasing the level of FOXP3 in regulatory T cells, we can avoid the regulatory T cells switch to an effector function, but also that they maintain their function once they have been transferred in vivo. We know that regulatory T cells are very important for immune balance, and we believe that by improving the persistence of regulatory T cells in the disease that Quell is planning to target, this will have a beneficial effect in the long term.